Um, I'm here to present an exciting ongoing project by my PhD student, Yojin Kim, in my lab at Georgia Tech in close collaboration with Dave Zhao and Heesun Han at the University of Illinois. So as spatially resolved transcriptomics or SRT technologies become popular, we're going to want to start comparing samples. And just as the original transcriptomics technologies immediately led to the search for differentially expressed genes and shared expression patterns. So as we think of this upcoming challenge, we wondered if it is possible to define an intrinsic coordinate system for spatial transcriptomic samples, just as one uses intrinsic coordinates to describe a car or an early embryo. Let me set up my laser pointer real quick. Okay. So such coordinate systems can simplify the comparison of multiple samples, allowing biologists, for instance, to understand how mutations influence spatial gene expression or temporal gene expression. The big question for us then was, is there a general purpose way to define a coordinate system for spatial transcriptomic samples? Give me a moment. Okay. So an obvious way is to use space itself, that is the x, y, and z coordinates. But this won't work because these coordinates are not comparable across samples. So the idea we had is this. Let's look upon gene expression as an encoder of spatial location. A classic example of this is how the expression of a few genes encodes the anterior, posterior, and dorsoventral position of a cell in the early Drosophila embryo. Since gene expression is comparable across samples, maybe it will provide a generalizable coordinate system for the tissue. So from this core idea, we developed a computational model to extract spatial coordinates out of gene expression profiles. We call this Space Express. Specifically, we wanted to learn a function f that maps the expression profile of a cell to its spatial location. In particular, the function f should match the expression vector g of a cell C to its location vector x. Even more precisely, the function we train maps the g of C to a low-dimensional vector p of C that captures the location xc of the cell. This vector pc can be considered as an embedding of the cell's location. And the dimensions of PC are going to be the coordinates of the cell in the desired coordinate system. So we want the embedding P to capture this, the location X, right? That is, the distance between the embeddings of any two cells, Ci and Cj, should be close to their spatial distance, Dij. We explored a number of different loss functions to implement this idea and found this particular function called Kamada-Kawai layout used in network layout settings to be the most useful. The model itself is a simple neural network that maps the gene expression vector GC to its embedding PC so that the pairwise embedding distances match spatial distances. And that's it. So we're now ready for some tests. We tested the idea first with a synthetic data set, which crudely mimics the Drosophila early embryo, with some genes that have an anterior-posterior pattern, some have a dorsoventral pattern, some a combination of those, and most genes are not patterned, so not spatially patterned. We then asked for this idealized synthetic sample, how well is the expression to location mapping captured by the model? To answer this, we mapped the cells from the SRT data set, this synthetic data, to their embeddings using Space Express. Then we used the known locations of the cells to construct a neighborhood graph and constructed a similar neighborhood graph from the embeddings and compared the two graphs using a Jaccard similarity score. And the answer in this case was a rather impressive 73%. This is a visualization of 4 of the 8 dimensions of the learned embedding vectors. We see that these 2 dimensions here they quite nicely captured the DV and AP axis of the synthetic embryo, which are indeed the intrinsic coordinate system of this synthetic data set. Of course, there are several other tools for embedding cells using their expression and location. So we compared SpaceXpress to a selection of these, including Stagate, Setter, and GraphST, and found SpaceXpress embeddings to capture space more effectively than these tools. This may be expected since the model was optimized specifically for capturing space. In a second synthetic data set, we simulated expression variation that is more of a clustered type rather than continuous gradients, with 9 spatial clusters shown here as a grid of cells. Each cluster marked by a marker gene, while 91 other genes are not spatially patterned. Here also, SpaceXpress showed a clear improvement over other embedding methods, and a PCA plot of the SpaceXpress embeddings shows that the original spatial structure is captured rather well in the learned embeddings. We next tested the method on 3 different real data sets spanning 3 different species and technologies. These included the popular 10x Visium dataset on the human dorsolateral prefrontal cortex, a star map data on the mouse cortex, and a stereoseq data on late embryos in Drosophila. And in each case, the accuracy of SpaceXpress embeddings capturing space was in the ballpark of the leading method, either Stagate or GraphST. Now it's time to remind you that SpaceXpress learns embeddings under a severe constraint, that they be pure mathematical functions of a cell's expression alone. This is different from all the other competitors. The reconstruction accuracy assessed in these slides is not our ultimate goal, because if it was, then we could use SpaceXpress on cellular transcriptomes that reflect the cellular neighborhoods and then get a far more dramatic improvement in accuracies. So these are numbers with SpaceXpress where we use a cell's transcriptome along with its neighbors' transcriptomes. But we stick to the baseline SpaceXpress model that respects the hard constraint of looking only at the cell's own transcriptome when computing embeddings. And then we examined the generalizability of these learned embeddings, since it's really the embeddings themselves that we are interested in for comparative analyses. For this, we first trained the model on one sample, in this case, human DLPFC, learning these particular dimensions, among others, and then applied the learned model to embed cells in a different sample from a different donor while ignoring spatial information completely. So we did not look at the locations of cells in this. And we saw that the embedding dimensions mirror the analogous directions in the training sample. Making this assessment more objective, we noted that cell pairs in different layers, there are all these layers in this, right, annotated, are more separated in the embedding space. This is a separation in the embedding space if their layers of origin are more separated in the real space. Also, cell pairs in the same layer are more separated in the embedding space if they are more distant in the physical space. We saw the same generalizability of the embeddings in a completely different data set where we trained SpaceXpress on one slice of the Drosophila embryo, slice 8, and tested it on a different slice whose spatial information was not examined. So these 2 examples illustrate that SpaceXpress can extract spatial information from a non-spatial single-cell data set once trained on an SRT data of a comparable tissue without mapping to any reference tissue. Having convinced ourselves that the learned embeddings really do represent an intrinsic coordinate system of a tissue, we next looked at comparing spatial patterns between tissue samples. You're well aware of methods that find spatially patterned genes in one sample, but it's not clear how we can rigorously detect changes in those spatial patterns of a gene between samples. A simple idea was to use each learned embedding dimension from SpaceXpress as a coordinate axis in which expression may change between samples. To illustrate this idea and to test the statistical method I'm going to talk about, let's start with a well-controlled synthetic data set 2 synthetic early embryos with very similar transcriptomes overall, and make the expression pattern of one of the genes slightly different between these 2 samples, right? Synthetic data. And ask if we can devise a test to detect such differential spatial expression. The testing procedure starts with learning space express embeddings on the 2 samples jointly, and then modeling the gene's expression as a function of each embedding dimension using a spline-based model. And then using a statistical test, the Walls test, for significant difference between the 2 embryos to obtain a p-value for differential spatial expression. We use this approach to detect differential spatial expression between whole brain Murfish data from honeybees in 2 different behavioral states, forager and soldier. These colored structures here, 4 in each, are the 4 mushroom bodies in each brain that play major roles in sensory integration and memory formation in these honeybees. Here's an illustration of one of the learned dimensions, the space express embedding dimensions, when embedding cells from the purple mushroom body in the forager and the soldier. So the dimension goes kind of high towards this part of the mushroom body. And this is an example of a gene that was found to be significantly differentially spatially expressed in the coordinates of that embedding dimension. As you can see from this more obvious plot where that embedding dimension is the x-axis and the expression is the y of this particular gene is the y-axis, and every dot is a cell. This is another example of a gene that was detected as differentially spatially expressed in each of the 4 mushroom bodies, so red to red, purple to purple, etc., between forager and soldier brains. For instance, in this mushroom body, the red mushroom body from this plot, the gene is clearly more expressed in the middle part of the mushroom body, the axial part, only in the forager brain and not so in the soldier brain. So in summary, the take-home messages from this talk are that we present a method to learn an intrinsic coordinate system for our tissue from spatial transcriptomics data. The learned coordinate system is generalizable and hence more reliable and robust. The tool can extract spatial information from non-spatial data without explicitly mapping to a reference sample. And we have the first rigorous approach to quantifying differential spatial expression. The work was done almost entirely by Yojin Kim with help from Zejun Wu and Anurendra Kumar in the lab, and in collaboration with Dave Zhao and Heesun Han at UIUC. I also thank my funding agencies and foundations. I would be happy to take questions. Is the moderator here yet? Sorry, I heard. Okay. Any questions? Yes.
So, because you're not considering space, when you're doing comparisons between mushroom bodies, are you taking any like morphological differences into consideration? Or I guess maybe I misunderstood that.
No, that's a great point. No, we're not we're not accounting for any morphological differences. And if there are differences, we're hoping that the coordinate system will learn, because it's learning kind of separately in the 2 mushroom bodies. If one is a distorted version of the other, it will learn that automatically. Like we saw in the DLPFC example, where one donor, the layers were kind of straight and the other, they were kind of bent. Those kinds of changes will be automatically taken care of. But if there are major structural differences, then we haven't gone to that part yet. That's a good point. Any other questions? Okay.
I think it's wonderful what you're doing. If you refer to lots of Drosophila and so on, so if you believe the work of John Reinitz, you will perhaps believe that so-called spatial patterns of gene expression don't really represent space, but they represent something a little bit different. Are you familiar with Reinitz?
Well, a fair bit, yes.
Reinitz claims that certain so-called spatial patterns of gene expression move over time, so they don't actually represent absolute space. I'm just curious if you want to comment on whether.
If you're referring to the Nature paper on the dynamics of the spatial patterns, Reinitz's paper, then this is for any. There's no temporal aspect or dynamics included in the model yet. So, but that paper was talking about how spatial patterns evolve during developmental time, rather than question the space itself. So let's talk offline if there's something else you're alluding to here.